Hi, this is Mrs. Hahn. I'm going to be going over the photosynthesis lab with you. Before we begin, I just want to make sure that you're clear about what photosynthesis is. So here we've got the reactants or the ingredients that photosynthesis requires, and that's sunlight, carbon dioxide, and water. And the plant uses those three reactants to make glucose or sugar and oxygen. And this picture shows that kind of in reverse, but the plant is making glucose for its own food. So we don't really have to feed plants, they can make their own food. And as they're making glucose, oxygen is a byproduct that's released into the air. So the more glucose they make, the more oxygen is released. And so that is photosynthesis. We're not going to be measuring glucose in this lab, but we're going to be measuring oxygen. So the more oxygen we see, the more photosynthesis is happening, the more glucose is being made. And that's how we'll be able to measure it. So let's take a look at the lab. When you open this lab, as you pop it out, you'll have the option to open it with Cami. And so if you're going to print it out, that's fine. If you're going to annotate on your computer, use Cami. If you would like Google Docs, this usually messes up the formatting. So I would ask that instead of clicking on Google Docs, you're going to use this link right here to get the Google Docs version, which is what this looks like. All right, so let's go ahead and open up Google Doc version and our lab. And I'll just kind of walk you through the different sections. So here's our warm-up. You can just sort of answer these questions, what you think these bubbles are, and then it's going to say select the bar chart here, and notice the oxygen production, move the light intensity back and forth, okay. And you can see that the bubbles go away, and then now there's more bubbles as we increase the light intensity and decrease it. And so it seems like these bubbles are oxygen bubbles. Okay. And then we're going to experiment with the temperature slider and see if that affects oxygen. So you're going to move it up and down and see, does that affect oxygen? If I move it warmer, what happens to the oxygen flow? If I move it colder? And then how does it affect the CO2? How does CO2 affect oxygen? Okay, so let's see, more CO2, less CO2. So right here, as you decrease the CO2, it looks like it decreases the oxygen flow. Okay. But after a certain point, it doesn't really seem to affect the oxygen flow. But if you have too little CO2, then you're not going to have as much oxygen. Okay, and how does oxygen production relate to the rate of photosynthesis? So what do you think, if there's more oxygen being produced, what does that say about how much photosynthesis is happening? And if there's a little bit of oxygen being produced, what does that tell you about the rate of photosynthesis? And if you remember our picture, this one. So if there's more oxygen being produced, then what do we know about photosynthesis? And if there's less oxygen being produced, what does that tell you about photosynthesis? All right, so let's go ahead and look at activity A. Let's go, we're still at the bar chart, and now we're going to turn on numerical values. Okay. Here is your hypothesis. We've got carbon dioxide, water, and light, and that should probably be an arrow, makes glucose and oxygen. Okay. And so here, what light intensity and CO2 level do you think will maximize the rate of photosynthesis? So remember, we want to see, right now, the only way we can see how much photosynthesis is happening is by measuring the oxygen flow, because we can't in this experiment, we can't really see how much glucose is being made, but we can see how much oxygen is being produced. So the more oxygen that's being produced, that tells us more photosynthesis is happening. So we want to see what should the light intensity, the CO2 level, and the temperature be to get the most oxygen produced. And so you're going to kind of fiddle with these different levels to see at what level will I have, what is the ideal light intensity, what is the ideal CO2 level, and temperature. So let's go ahead and try temperature. So what would be the ideal temperature? Well, if I go too high, then the oxygen level goes too far down. If I go too low, it's also going to go down. So it seems like the highest amount of O2 that's going to flow is right around 29 degrees Celsius. And it's 37.8, around 27. So that looks like an optimal temperature. 37, 27. Yeah, around 27. Okay. And you're going to do the same thing with the light. You're going to see at what light intensity does it seem to be optimal for the most amount of oxygen production. Okay. At what light level is it really going very high? Okay. And then same thing with CO2. So then we're going to answer these questions. Activity B, we're going to see, does the color of the light affect the oxygen production? So let's take a look at the color of the light. So we can go to the color tab, we've got the bar chart, and we're going to set the temperature to 24. We're going to set the light intensity to 90. And the CO2 level to 1000. Okay. And we're going to read a little bit about this pigment called chlorophyll, which absorbs light energy and converts energy into one that the plant can use. And we want to figure out what color of light is best for photosynthesis. All right, so now we are going to move this light wavelength back and forth and see how it affects photosynthesis. Now, how do we know which one of these to pay attention to for photosynthesis? Well, we want to look at oxygen production, because the more oxygen that is being produced, we know more photosynthesis is happening. We know more glucose is being made. So I'm going to go ahead and see here. So it looks like I'm making a lot of oxygen at this wavelength. Ooh. So these higher wavelengths, I see more. Not a lot here. And then a lot here. Okay. So it looks like different colors, maybe around the ends, but not really here in the middle, the green, and then more over in the orange and red. Seem to be ideal. Okay, so now we're going to gather some data. We're going to set it at 400. And then we're going to go ahead and record, go to the table. Let's see. Do we need to change anything on the light intensity? I don't think so. Okay, we're going to go ahead and record the data every 20 nanometers. So here we're going to go ahead and hit record data, and then we're going to move the wavelength 20, and we're going to record data. Move it 20 and record data all the way to the end. And then you're going to be able to see which colors were absorbed best by the plant. I'll do a couple more just so you can see what this looks like. I won't do the whole thing. Okay. But you're going to go all the way. You're going to go all the way to the end. And then you're going to hit graph. So mine is not finished, but yours will be finished. And then we're going to hit wavelength. So you can see that there's a lot of oxygen being produced here, but as we get closer to this wavelength, oxygen is not being produced as much. So this area, these colors are absorbing the light better than what's coming up here, these colors. So when the plant is absorbing the light, it's producing a lot of oxygen, which is why it's very high here. But as it's going down, and it looks like not a lot of oxygen is going to be produced over in this section, that is not going to be absorbed. And one thing you know about plants is that they're green, and so the reason that plants are green is because they're reflecting light. So that actually makes sense. If they're reflecting light at this wavelength, they're reflecting the green, and that's why we see it as green. And when they're reflecting, they're not absorbing that light. Okay, let's go back to the white tab. We'll go back to white light, and then we're going to set the temperature at 24, light intensity at 50, and CO2 level to 200. Okay, and now we're going to see what the limiting factor is. So let's say you're making bread and you only have a certain amount of sugar. So that's your limiting factor, because you can't make any more bread once you run out of sugar. Same thing if you have only a certain amount of flour, then you can keep making bread until you run out of that flour, and that flour becomes the limiting factor, even if you have tons of all the other ingredients. So whenever something is in short supply, that becomes your limiting factor. So we went ahead and set these, and you're going to go ahead and see what would be the limiting factor of these different components. So we're going to go to bar chart. And again, we're going to look at our oxygen, because this is telling us how much photosynthesis is happening by the amount of oxygen being produced. All right, so we're going to go ahead and move the temperature slider up and down and see whether we're able to increase oxygen production. So I'm going to move it up. And right now the oxygen is at 23.4. So am I able to move it? Nope, it goes down. Okay, so basically whatever I do, it's not going beyond what it was, 23.4. So it is not, I would put no here. It is not a limiting factor. Next, I'm going to move light intensity and see if I'm able to increase oxygen production by moving the light intensity. So again, it's at 23.4. I'm going to move it around to see if I'm able to increase it. And then I'm going to move it back. Okay, so you're going to go ahead and answer those questions. And then you're going to try and find the limiting factor here. So let's go ahead and do the first one. So if it's 25, light intensity is 60. And CO2 level is 700. Okay, let's go ahead and see which one of these is able to increase the oxygen beyond 48.1. So nope, this one is not able to move it any higher than 48. Ah, light intensity goes up a little bit. 49. Okay, so light intensity can increase it a little bit. And CO2 level, nope. So my limiting factor would be, would be the light intensity, because the light intensity can affect can increase the photosynthesis rate if I change the light intensity. So that's my limiting factor. Okay, I hope that wasn't too confusing. If you have any other questions, feel free to email me, and good luck.